Hi everyone, my name is Hayden Glenn. I currently go to Winston-Salem State and I am their Astrobotany Lab program. And today I'll be talking about crop yield assessment and on-Earth regolith and space crop data analysis. So first, before we start, why, like a little bit of background, why is this important? So as of April 2022, as of April 2020, there's only been 22 peer-reviewed published papers on lunar crops. And of these 22, only 2 research studies have used sterilized stimulant, and the other 4 of those have used the same regolith types. And throughout these 22 reviewed papers, there's been no standardized methodology throughout any of them. So when we say basically, if there's no standard methodology, it generates unreliable data. So kind of going through our steps and what we decided to do. So here you can see our stimulant regolith types that we used: dusty, frosty, lunar highlands, and lunar mare. And these are the 3 crops we used as well. So we have tomatoes, mainly for their water content, wheat because they're fast flowering, and also, oh, sorry, I was reading the chat. I thought that was for me. And also, however, wheat does not have a lot of edible biomass, but because it flowers fast, it's a good crop to use. And then lettuce, because of its water content, as well as many studies talking about lunar crops, have used lettuce as well. And we broke it down into 4 phases. So here you can see our step 1 and results of that. So we decided to not use frosty and dusty stimulants because their anoxic pockets levels were too high. And so we decided that it wouldn't be great to grow the crops in because of that reason. So we decided to go ahead and just move through with the lunar mare and the lunar highlands. And here you can see the results of that. Oh, sorry. Here you can see the results of that. So we basically mapped out germination rate, total biomass, daily MAR, and edible biomass. And here we use the LMS. You can see LMS, LHS, and controls. And relatively all of them were around the same in comparison to the control. So basically, so after just kind of going through all the steps in our results, so we decided to only use 2 of the 4 original regolith types due to the anoxic pockets, the high anoxic pocket values in the dusty and the frosty. And then we can see the first generation regolith seeds were viable and they reached seedling stage, but due to COVID, we cannot really continue with that. We started that right before the shutdown. And then all the crops presented stress, and that was mainly due to the phytohormone production. And we think that's linked to the oxygen exchange rate, so the anoxic pockets, and also the low nutrient concentration. Because in the regolith, there's no ecosystem of bacteria, obviously, so there was a lot of stress on the plants to try to grow. But the specific phytohormones that we saw increased levels of stress in were the ethylene, auxin, and cytokinin. And these hormones influence lateral root formation, shoot formation, and early flowering of the tested crops. We also saw cell enlargement and division observed under the anoxic stress. And the cell enlargement was mainly in the lettuce leaves, but when there's higher cell enlargement, that equates to higher transpiration levels. So, and this is a stage we're basically working on right now. So because there's no microbiome in the regolith, we decided to make our own. So this is our gelatinous cube concept. And basically what it is would be agar-agar that you see in like a lot of chemistry and microbiology groups. And the 3 bacteria that we were mainly focusing on was Azospirillum, Rhizobium, and Pseudomonas. And we wanted to introduce those into our regolith / gel cube concept. And you can see, like, when we did an evaluation of the root exudates, we saw that there was needs for sugar, osmolize, and isoflavones, but we weren't able to test the last 3 at the bottom. So kind of going with what we decided to do, we had a regolith microbial recipe, which I was talking about in the last slide. You can see based on our graphs, and then we had a control, and then obviously we measured it against the regolith. And this time we decided to use soybeans and lettuce. So we both saw in the soy, and we call that soy and let one in our samples, we saw higher levels in total biomass and edible biomass when compared to the pure Regulus samples. There was also higher levels of oxygen and gibberellins in the soy and let one levels compared to the control in the oxygen or Regulus samples. And we think that might be due to the introduction of our microbial recipe, bringing in more nutrients and also bringing in more hormones. And then overall root architecture, the Regulus samples have more basal root area when compared to controls. So basically overall, we were able to see a definitely better increase, an increase in comparison to our Regulus versus our Regulus and microbial recipe. So far we think it's a success, but we have to continue finishing. And then here's a few pictures of me and my group. You can see some of us. In the top left is us at the NASA Kennedy Space Center. And then here's the picture and me in the blue with the guy in the orange shirt. We were in one of the chambers talking about different vegetables that they were growing. And here's a picture in the bottom left of just my whole group all together. And just want to thank the Space Grant for all their amazing help. We couldn't have done it without them. So we just want to continue to work with you guys and just be the best I guess we can be. So thank you.